My name is Mark Murakami. I'm a physician scientist at the Dana-Farber Cancer Institute in Boston and work with Drs. Reed Merriman and Philippe Armand in clinical trials and translational research in follicular lymphoma, supported by the FLF Academic Clinical Trials Program, as well as a translational research program grant. Support from the FLF has, first of all, allowed us to accelerate our translational research in association with our clinical trials. There are several individual sub-projects in our grant that where support from FLF has allowed us to move forward more quickly. It has allowed us to choose the right technologies and assays, not just the ones that were most readily available or the least costly. So I think it's allowed us to be more effective earlier with our translational research. That's number one. Number two, the support from the FLF, both on the trials level as well as on the science level, has allowed us to establish a larger scale of research that has given us some credibility, that has actually allowed us to leverage support from the FLF into procuring additional support for other trials and other scientific endeavors. So we're really excited that in addition to the trial that the FLF is supporting, we have another very similar trial in follicular lymphoma using immunotherapy, and we actually have a third trial that just recently opened. And these three trials are distinct but complementary, and we're applying some of the same scientific approaches across all three trials, which is very advantageous. I would say that one thing that I always like to highlight is that we're very fortunate at the Dana-Farber Cancer Institute to have a very cohesive group with very close links between the clinic and the laboratories. And so we receive amazing support from Dr. Philippe Armand, who's the PI of our Academic Clinical Trials Grant, and we have excellent collaboration on the laboratory side with other investigators like Dr. Aaron Perry and Dr. Reed Merriman, with whom we work very closely on early phase clinical trials. Scientifically, I think what distinguishes us is that we have a thematic focus in minimal residual disease, and that refers to the small number of lymphoma cells that remain despite the eradication of 99.9% of the cells with treatment. And so we're very focused on deeply interrogating those few remaining cells so that we can identify vulnerabilities that could be amenable to novel therapies in the future. If I had to highlight one singular goal for this next year, it would be that we have already established a very nice system for defining sensitivity of different lymphomas to different drugs, including immunotherapies like bispecific antibodies. And we're very optimistic that within the next year, we will have been able to evaluate enough lymphoma biopsy specimens that we will have some data supporting at least a couple of very promising combination partners for bispecific antibodies that we'll be testing very intensively over the next 1 to 2 years, and hopefully in the longer term future, advance into early phase trials.